Parkinson's disease, also known as PD, was discovered by James Parkinsons, an English surgeon, paleontologist, and neurologist. He described it as paralysis agitan. In 1817, he wrote an essay on the shaking palsy, which he described as involuntary tremulous motion which lessened muscular power. Dr. Parkinson treated himself and 6 other patients for Parkinson's disease. Through these treatments, he distinguished the difference between resting tremors and tremors with motion. Resting tremors are tremors when you're sitting at rest and your body is shaking. Tremors with motion are when you're moving throughout the day and you can still feel your body moving. 60 years later, a French neurologist and professor of anatomical pathology named Jean-Martin Cartet renamed paralysis agitans to Parkinson's disease, named after the founder and discoverer of Parkinson's, James Parkinson. Charcot is known as the founder of modern neurology. He studied Parkinson's between 1868 and 1881. He discovered the distinction between rigidity, weakness, and bradykinesia. Bradykinesia is slowness in execution of movement. Charcot received the first European professional chair of clinical diseases for the nervous system in 1882 for his discovery of the distinction between rigidity, weakness, and bradykinesia. April is known as World Parkinson's Awareness Month. April 11th is World Parkinson's Awareness Day due to the fact that James Parkinson was born on April 11th. J.W.S. van der Waals, a Dutch horticulturalist, had Parkinson's disease and honored Dr. Parkinson's with a flower. The flower is called the red tulip, and they are the official flower for Parkinson's disease. This flower received Award of Merit in 1881 from the Royal Horticultural Society in England. Parkinson's disease is a chronic and progressive disease of the nervous system. Chronic being that it persists over time, progressive being that the symptoms grow worse over time. PD is the second most prevalent neurodegenerative disorder. Neurodegenerative is a term used for conditions which primarily affect the neurons in the human brain. Neurons are building blocks of the nervous system. The nervous system contains the brain and the spinal cord. PD is characterized by bradykinesia, resting tremors, and rigidity due to the loss of dopaminergic neurons within the substantia nigra section of the ventral midbrain. Dopaminergic neurons are the main source of dopamine in the mammalian central nervous system. Dopaminergic systems have been divided into nominal systems. The main system, the nigral striatal pathway, plays an essential role in the control of voluntary motor movement. Substantia nigra is part of the extrapyramidal system, which is part of a motor system involved in regulation of movement. The substantia nigra lies in the midbrain immediately dorsal to the cerebral peduncles. Cells within this system produce dopamine normally. Dopamine passes to the nerve terminals in the striatum where it is released as a transmitter. If there is no transmitter produced, it involves in the crippling disorder of Parkinson's. Transmitters help control movement and coordination. In most cases of Parkinson's disease, protein deposits called Lewy bodies appear in dead or dying dopamine-producing neurons. Cases without Lewy bodies are sometimes referred to as Parkinsonism instead of Parkinson's disease. It is unclear whether Lewy bodies play a role in killing nerve cells or if they are part of a protective process. This imbalance causes the symptoms of Parkinson's disease. When dopamine and acetylcholine are equal, this causes good motor movement. The causes of Parkinson's disease are not known. However, we know that in people with Parkinson's, neurons in the substantia nigra slowly die off. This causes less dopamine to be made. Nigrosim 1 is the largest proportion of neurons affected in PD. In healthy people, nigrosim 1 is supposed to be oval, While it is absent in patients, it sometimes looks like a swallowtail. A swallowtail is a tail of a bird. Dr. Schwartz is helping in diagnosing Parkinson's disease by simply just looking at an MRI to notice if there's swallowtail present. People diagnosed with Parkinson's disease have many symptoms. One symptom is the loss of motor skills. Motor skills are trouble walking, sitting, talking, or completing simple tasks such as your body is shaking, speaking. Usually, some people have urinary problems. PD attacks people over the age of 60. Doctors have been studying the tremors. They're divided into two separate categories: major tremors or minor tremors. Major tremors are constant throughout the day, leaving large involuntary movements of the limbs, which causes rapid weight loss and inability to sleep or eat. Minor tremors tend to disappear throughout the day. During examination, rigidity can be seen by the doctor. It occurs in the neck, shoulders, hips, and the wrists and ankles. Postural instability is a loss of balance and is in extreme cases. Usually, this is why people with Parkinson's disease are very prone to falling. About 60,000 Americans are newly diagnosed each year with Parkinson's disease. More than 1 million Americans are affected at one time with Parkinson's disease. People with parents and siblings that have Parkinson's disease are 2 times the likely chance of developing Parkinson's disease. Most people affected with PD are not aware of any relatives with the condition, but in a number of families, there is a family history. When 3 or more people are affected in a family, especially if they are diagnosed at an early age, we suspect that there may be a gene making this family more likely to develop the condition. Approximately 15% of people with Parkinson's disease have a family history of this disorder. These familial cases are caused by mutations in the LRRK2, PARK2, PARK7, PINK7, or CNCA gene, or by alterations in genes that have not been identified. Mutations in some of these genes may also play a role in cases that appear to be sporadic. In some families, alterations in the GBA, SNCAIP, or UCHLI gene appear to modify the risk of developing Parkinson's disease. There are 2 categories of genes: casual genes and associated genes. Casual genes actually cause the disease. A casual gene alone, without the influence or other genes or environmental factors, guarantees that a person who inherits will develop Parkinson's disease. Only 2% of people with Parkinson's disease have casual genes. Examples of these genes include SNCA, PARK2, and PARK7. Associated genes do not cause Parkinson's on their own, but increase the risk of developing it. A person may have these genes and never develop Parkinson's disease, while people who do not have these genes can still end up being diagnosed with Parkinson's. Examples of these genes are LRRK2, GBA, SNCAIP. The GBA gene has the official name of glucosidase beta acid. The GBA gene provides instructions for making an enzyme called beta-glucocerebrosidase. This enzyme is active in lysosomes, which are structures inside cells that act as recycling centers. The location of this gene is 1q21. GBA mutation carriers have an increased risk of developing Parkinson's disease or Parkinsonism. The LRRK2 gene has the official name of leucine-rich repeat kinase 2. The LRRK2 gene provides instructions for making a protein called dardarin. The LRRK2 gene is active in the brain and other tissues throughout the body, but little is known about this gene or the dardarin protein. The location of this gene is 12q12. Researchers have identified at least 20 LRRK2 mutations in families with late-onset Parkinson's disease. These mutations replace one amino acid with another amino acid in the dartarin protein, which affects the protein's structure and function. It is unclear how LRRK2 mutations lead to Parkinson's disease. The PARK2 gene is one of the largest human genes that provides instructions for making a protein called parkin. Parkin plays a role in the cell machinery that breaks down unwanted proteins. Researchers have identified more than 100 PARK2 mutations that cause juvenile Parkinson's disease and some adult-onset Parkinson's cases. Some mutations lead to abnormally small parkin protein, which is non-functional and degrades rapidly. Levodopa, also called L-DOPA, is the most commonly prescribed and most effective drug for controlling the symptoms of Parkinson's disease, particularly bradykinesia and rigidity. Levodopa is transported to the nerve cells in the brain that produce dopamine. Cyme is made of levodopa and another drug called carbidopa. Levodopa enters the brain and is converted to dopamine, while carbidopa increases its effectiveness and prevents or lessens many of the side effects of levodopa, such as nausea, vomiting, and occasional heart rhythm disturbances. It is generally recommended that patients take Cyme on an empty stomach at least 30 minutes before or 1 hour after meals.